So welcome to the Brain Prize webinar on learning and memory. This year's Brain Prize was given for computational and theoretical neuroscience. And today we have three speakers who are experts on modeling learning mechanisms and synaptic plasticity. The first speaker is Nicolas Brunel, who has a background in physics, and his particular expertise is on trying to model the mechanisms for learning and memory at both the synaptic and the network level. He collaborates with experimental groups analyzing data, and he uses methods from statistical physics. So Nicolas, could you put up your screen and let us know? By the way, he was until recently at the Duke University. He's moved recently to the University of Milan on the faculty.
So can you see my screen?
Yes.
Okay. So you see only the PowerPoint presentation and nothing else?
We're fine. Sure go okay. Ahead.
Okay. All right. Well, first, thanks, Terry, for asking me to be a participant of this webinar, and of course, congratulations to all three of you for the Brain Prize. I think it's a highly deserved recognition of the incredible contributions that you've had to the field, and also I think it's a recognition more generally of the impact that the theoretical and computational neuroscience field has had on neuroscience as a whole, and it shows the deep impact that computational neuroscientists have had on the field. And in particular, it's had a strong impact, I think, on the field of learning and memory, so the topic of today's webinar. And by the way, this is also a topic on which all three recipients of the Brain Prize have had pioneering contributions that are too numerous to mention. In fact, the understanding learning and memory is really one of the areas of neuroscience where there is a very acute need for computational modeling tools, because and in particular because of the multi-scale nature of the problem. So learning induces modifications of synaptic strength in a multitude of synapses across multiple brain areas. And to understand how these synaptic modifications lead in turn to modifications of dynamics of neural circuits and in terms of modifications of behavior, we need mathematical models that allow us to bridge these two levels. And theorists have used a multiplicity of approaches to try to understand better the mechanisms of learning and memory, starting from different starting points. So in this presentation, I'd like to briefly present three different projects for my group where we've tried to advance our understanding of this phenomena using different starting points. So in the first part of the presentation, the starting point will be in vitro data that allowed us to build a mechanistic model of synaptic plasticity that allow us to reproduce the data and also make experimental predictions. In the second part of the talk, the starting point will be in vivo data, and I will show how in some cases we can infer synaptic plasticity rules from this in vivo data. And finally, in the third part of the presentation, the starting point will be optimality principles. And we'll try to understand how we can derive the statistics of synaptic connectivity networks that optimize information storage capacity. All right. So most of what we know about synaptic plasticity comes from in vitro studies. And these studies have in particular clarified what are the requirements for plasticity to occur. In particular, one of the requirements is the entry of calcium ions through NMDA receptors that are typically opened when there is coincidence between pre and post synaptic activity. These calcium ions then trigger a cascade of biochemical reactions that lead in turn to either potentiation or depression of synaptic strength. And we know also how these calcium transients look like, thanks to experiments such as the ones shown here, in which these calcium transients triggered by pre or postsynaptic spikes have been measured. So these experiments have suggested that a good starting point for building a mechanistic model of synaptic plasticity is to consider that calcium is the primary variable that drives plasticity. And so these models were pioneered by Harel Schuval and collaborators. And together with a graduate student, Michael Grobner, we introduced a highly simplified version of a calcium-based synaptic plasticity model, which is shown here in this panel. So in this panel, we have a synapse connected to neurons, pre- and postsynaptic neurons, and spikes in both pre- and postsynaptic neurons trigger calcium transients of amplitude C pre and C post respectively, and these transients decay exponentially with the time constant tau Ca. In the model, we add two thresholds, one threshold for depression, theta d, and a threshold for potentiation, theta b. And the idea is that in this model, whenever the calcium concentration exceeds the potentiation threshold, there is an increase in synaptic efficacy with rate gamma p, while when the calcium concentration is in between these two thresholds, then the efficacy will decrease with a rate gamma d. So the advantage of this model is that it's very simple. All the parameters of the model are basically shown on this slide. And we can explore all possible behaviors of this model analytically. And so that's what we did with Michael Grobner. And in particular, we focused on experimental protocols that have been used extensively, in particular the spike time independent plasticity protocol. So we showed that when we use the spike time independent plasticity protocol, this model can exhibit a diversity of so-called STDP curves that characterize the dependence of plasticity on the time difference between the pre and the postsynaptic spikes. And these dependence on these different curves can be obtained by varying the amplitude of calcium transients C pre and C post relative to this threshold theta d and theta p. In particular, on the top left, we see here the classical STDP curve that reproduces the curve that was recorded by B and Pu in hippocampal cultures, but we can find also other types of STDP curves. And interestingly, this model is able to recapitulate all the possible types of STDP curves that have been characterized in vitro, to my knowledge. And this can be done by simply varying these two parameters, C pre and C post, or equivalently theta d and theta p. In addition, the model can also reproduce the dependence of plasticity on firing rate. This is just one example of the model fitting data recorded from Jesper Sjöström and collaborators, where what Jesper did is to look at how plasticity depends on the frequency of the spike doublets at different values of delta t. And we see that while at low frequency, the plasticity dominated by depression, and there is no significant potentiation. At high frequency, the reverse occurs, and there is potentiation at all possible delta t. Plasticity becomes independent of delta t in that limit. So this model can also make experimental predictions. And one prediction is that, so in the model, the STDP curve should strongly depend on the amplitude. We know in the model that STDP curves strongly depend on the amplitude of calcium transients. And so that predicts that STDP curves should strongly depend on extracellular calcium. So in particular, if we start at a high calcium concentration and we observe a STDP curve that has one potentiation window surrounded by two depression windows, so that means we are in this region, then we predict that by decreasing calcium concentration, we should first go through a transition where the LTP window disappears and we are left with only depression. And then a second depression should occur and we should end up in that lower triangular region where no plasticity occurs. And furthermore, investigating how plasticity depends on calcium concentration is also important because in most of the experiments that have been done so far, calcium concentration is pretty high in the range 2 to 3 millimolar, while values that have been recorded in physiological conditions indicate lower calcium concentration in the range 1.3 to 1.8 millimeter. So more generally, that this raises the question, how do STDP curves look like in physiological conditions? And so for this, we teamed with Yanis Englebert and Dominique Debarne from Marseille. Dominique is an expert of hippocampal plasticity, and they did recordings in hippocampal slices, and these are recordings of the classic CA3 to CA1 pathway. They induced plasticity of that pathway using a standard STDP protocol with spike pairs at low frequencies at a fixed delta T, and they used different calcium concentrations in these experiments. And what they found is remarkably in striking agreement with the prediction of the model. We see that at high calcium concentration, at 3 millimolar, we have a potentiation window surrounded by 2 depression windows. When we lower calcium concentration to 1.8 millimolar, we no longer have any potentiation and only depression remains. And finally, at 1.3 millimolar, we see that there is no significant plasticity at any delta T. And so this suggests that, in fact, at physiological levels of calcium, there should be no plasticity in this particular protocol. The model then suggested ways in which we can recover plasticity at physiological levels. And the prediction is that either by increasing frequency of the rings or by using bursts instead of single spikes, then we could recover plasticity. And both of these predictions were shown to be true in hippocampal slices. All right, so these results indicate that that plasticity in vivo could be quite different from what in vitro experiments have told us. So ideally, we'd like to observe plasticity in vivo experiments, but of course, it is much harder because we don't have direct access to synaptic strength as in vitro experiments. So back about 10 years ago, when I was in Chicago, we wondered whether it could be possible to infer synaptic plasticity rules using data in which the response of neurons to 2 different sets of stimuli is recorded. One set is a set of novel stimuli, and the other set is a set of familiar stimuli, with the idea that these familiar stimuli have been presented many times to the animal and therefore presumably have modified the synaptic connectivity. And so the idea is that by comparing these 2 different sets of stimuli, or the responses to these different sets of stimuli, we could perhaps infer something about the plasticity process that led to these differences. And in particular, we collaborated with both David Friedman from the University of Chicago and David Scheinberg from Brown University. And they both had recordings from IT cortex of primates. And in these recordings, they recorded the activity of neurons from IT cortex while they presented novel and familiar images to the monkey. And this is an example of one IT neuron from a monkey recorded from David Scheinberg's laboratory. And we see that when we compare the histograms of visual responses to novel and familiar stimuli, we see two things. First is that the average response to familiar stimuli is lower than the average to novel stimuli, and this is something that's been reported consistently by many investigators. However, we also see that the strongest responses are actually responses to familiar images, not novel. The question then is, what kind of plasticity rule could drive these differences in the statistics of neuronal responses to novel and familiar? So to answer this question, we use a standard rate model, and we generated novel stimuli in such a way so that the distribution of rates in response to this stimuli would match the empirical novel distribution, so the red curve here. Then we applied an unsupervised plasticity rule, delta Wij is in this model a function of the postsynaptic firing rate times another function of the presynaptic firing rate, f and g. So two functions, f and g. This will change the connectivity matrix. So now this particular stimulus is now considered familiar when it has modified this connectivity matrix. And then the idea is to find these functions f and g such that the distribution of rates in the model matches now the empirical familiar distribution after learning. And what we found is that indeed we could infer the rule delta wij, and we found that in most the IT neurons, in most IT putative excitatory neurons, we found that this plasticity rule is Hebbian in the sense that both f and g are increasing functions of the post and presynaptic rates, respectively. And this is an example for this particular neuron. We see that the function f here is well described by a sigmoidal function. So note that, of course, one example in the family of rule would be the covariance rule that was, in fact, introduced many years ago by Terry. But in fact, here there are several differences with the covariance rule. First, the function f is strongly nonlinear, as we see here. Second, the average of the function f over the distribution of postsynaptic rates is negative. And this negative average that really leads to the decrease of average firing rates with familiarity. Third, this function is characterized by a high potentiation threshold. The threshold for potentiation is much higher than the mean firing rate, than the mean visual response. And the consequence is that the increase of firing rates with familiarity only occurs for a very small fraction of the best stimuli, the ones that initially led to the highest rates in this neuron. And together, these effects lead to a sparsening of representation with learning, which is with familiarity, and which makes a lot of sense for multiple reasons. And also, I would like to note that this plasticity rule that we inferred has a similar dependence on firing rates as in vitro experiments and also as the calcium-based model, as I showed a few slides ago. We've also studied the dynamics of networks with plasticity rules inferred from data, so plasticity rules of the type I just presented. And we showed in particular that networks with such plasticity rules can exhibit attractor dynamics. So they can essentially behave as a Hopfield type network, where each of the fixed point attractors is similar to the state of the network when the corresponding stimulus was shown in the first place, but not identical. It's actually different. And in fact, this model is similar to the Hopfield model, but in contrast to the Hopfield model, this model has both a learning rule and also distribution of firing rates in both visually driven states and also delay persistent states that are similar to data. And also, these models can have a diversity of attractor states. We can have either fixed point attractor states or chaotic attractor states, but that can still maintain a stimulus in short-term memory. All right, so now I'd like to move to the third and last part of this presentation, and in fact, I'd like to present you a completely different approach to investigating learning and memory in networks. So in the first two parts, we've really focused on synaptic plasticity rules that we've inferred either from in vitro data or from in vivo data. In the late 80s, Elizabeth Gardner, a statistical physicist, proposed a completely different approach. The idea was, instead of focusing on specific learning rules, the idea was to study the space of all possible connectivity matrices that perform a specific computation. And the kind of computations that have been studied is in particular storage of random input-output associations in feed-forward networks, or storage of fixed-point attractors, or sequences of states in recurrent networks. So in these models, there are two types of constraints on connectivity matrices. The first type of constraints are due to the storage of memories, right? So for instance, for each input-output association you want to store, there will be typically that will have the form of a hyperplane that separates a subspace for which the network actually performs correctly this input-output association, or does not in that shaded space. And so that's the first type of constraints. And of course, the more information you want to store in this network, the more you will be restricting the available subspace of weights. The second type of constraints in biological networks is due to our sign constraints, is due to Day's law. So these are these two hyperplanes that are indicated here. So what Elizabeth Gardner showed is that what you can do using methods from statistical physics is to compute the volume of this subspace when you assume patterns are random with specified distributions. And that in turn allows you to compute the maximal storage capacity of these networks, which is simply the point at which the volume will shrink to zero when you increase the number of associations or attractors that you want to store in these networks. So what we did then is to generalize this approach to rather have a closer look at the connectivity matrices on the subspace, and then to compute basic statistics of these matrices, right? So for instance, distribution of weights in these networks, joint distribution of weights in pairs of neurons in recurrent networks, distributions of motifs, etc., etc. And so this is just one slide to summarize the main results of this approach. What we find is that in networks that maximize storage capacity at a given robustness level, or vice versa, maximizing robustness for a particular storage capacity, then we find that the resulting connectivity matrices are sparse. So more than half of the synapses have exactly zero weights, and that's really a result of this, of Dale's law, of the sign constraint. Interestingly, the more robust storage is, the sparser the network, the higher the fraction of zero weight synapses. And we think that's consistent with observations in multiple brain structures, and in particular, the discrepancy that's been observed between potential and electrophysiological connection probabilities. And that has been noted both in cerebellar granule cell to Purkinje cell weights, where the connection probability could be 50% anatomically, but in practice, electrophysiologically, it's only 10%, and also in cortex, in cortex, in particular, layer 5, pyramidal to pyramidal weights, where the connection probability is much lower than what it could be anatomically. Second, we find that the theoretical distribution of weights fits pretty well the empirical distribution of weights. So this is an example in cortex, distribution of weights of layer 5 to layer 5 pyramidal cells. And this is a comparison between the model. And by the way, this is not a fit. This is a prediction of the distribution from the model that can be computed only with only two parameters, the connection probability and the mean weight. Third, we find that in recurrent networks storing fixed point attractors, we find that a prediction of an overrepresentation of bidirectionally connected pairs of neurons. And in particular, we see here the reciprocal connection probability as a function of the connection probability. And we see that these networks that are close to maximal capacity, they are approximately halfway between a fully symmetric network, which would be this diagonal line, and a random directed Erdős-Rényi graph, which would be this y equal x square line. And this is basically these different curves corresponds to the model, varying the robustness parameter and varying also the coding level. And this is basically the data measured in cortex is this red asterisk. And finally, there is also an overrepresentation more generally of highly clustered motifs in these networks, and that also reproduces multiple experimental observations in cortex. So finally, I'd like to mention all the people that have worked on these different projects, in particular, the project about the calcium-based plasticity rule was spearheaded by Michael Gropner while he was a graduate student in Paris, and then continued by Jonathan Aljadev. Jonathan is now a faculty at UCSD. The second part about the inferring plasticity rules from in vivo data was initially spearheaded by Sukhbin Lim, who's now faculty at NYU Shanghai, and then pursued by Ulises, who's now at Allen Institute. And we've also collaborated with a number of experimentalists, Yanis and Dominique from Marseille, David Friedman from Chicago, and David Chamber from Brown. This is a mistake here. This should be Brown. And then also other theorists, Yali, Vincent, Jean-Pierre, and Boris is an experimentalist working on cerebellum at INS Paris. Thanks for your attention.
Well, thank you very much, Nicolas. You covered a lot of ground there. But I think this really illustrates the close ties between theory, computation on one hand, and experiment on the other side. And it really is giving us insights into the statistics of what is measured in the brain regarding plasticity, regarding connectivity patterns. And so we have time for a few questions. And I want to start out by asking about STDP. I think you mentioned briefly that in vivo, one way to stimulate it might be with bursts.
Yes.
High frequency bursts. And I wonder if that could be related to the oscillations that are seen. So very ubiquitous, found in the cortex over a wide range of frequencies, from 2 hertz up to 100 hertz, you get these bursts. And I wonder if you think that they're connected at all to STDP.
Yes, well, thanks for asking this question. So in this model, plasticity is so the prediction from the model and the corresponding experiments in hippocampus is that in some conditions, plasticity would not occur with single spikes, but rather only with bursts. And in fact, oscillations would be a good way to bring spikes closer together. And I think that could explain why plasticity happens in some condition more easily in some frequency ranges. For instance, I think it's been shown in hippocampus that oscillations in the theta range tend to favor plasticity. So yes, I think it is one potential benefit of oscillations.
Yeah, that I think is a very interesting and intriguing direction. John Listman actually was involved in a lot of the early work in the hippocampus, and of course, he also had some ideas about the calcium thresholds.
Yes.
So, Juliana, do you have a question?
Yes, I actually had 2 questions. Thank you very much, Nicolas. That was a very beautiful overview of all the amazing work that you have done, and of course, so I wanted to ask about mainly the second part, inferring the plasticity rules from in vivo data. So I was wondering, can you or have you thought about extending the methods that you have to apply to individual neurons, not just population? So you showed, how you can go from one distribution in the context of images that are novel, then become familiar. But can one do the same if you have sort of the activity or the dynamics of individual neurons? And then related to that, even with the current framework, could you apply it to other data sets that are not necessarily applying to, the pre-post being novel versus familiar, but let's say monocular deprivation or some kind of learning that an animal and goes through like fear conditioning or even lever pressing or?
Yes, thank you for the questions. So first, I apologize for going too fast on that part, but actually the method that we derived actually can infer plasticity rules for individual neurons, because what we're really considering are the distributions of responses to novel and familiar neuron by neuron, and for each neuron we can actually infer its own plasticity rule. And we've actually find a big heterogeneity of plasticity rules, and part of it, of course, can be explained by the fact that we have finite numbers of stimuli that we're using, and also and of noise in visual responses. But we really can. This method allows us to derive plasticity curve for single neurons. The second part of the question, yes, I would very much like to extend this method to be much more general than just this specific context, and in particular to apply it to, in general, experiments where we can follow the activity of neurons as an animal learns something. So in fact, I'm currently involved in a project which is actually with people at Duke University that record activity in primate cerebellum while the animal is actually learning a task. And so the idea is to generalize this method to be able to infer plasticity also in these conditions. But yeah, it's too early to be able to present these results.
Can I just come back to the first part of the question? Let's say maybe not just firing rates, but let's say also correlations between the individual neurons. Yeah. So high order.
Yeah, no, so also I pushed under the rug all the assumptions that we had to make. So we ignored correlations between neurons. These are, by the way, just single unit recordings in IT cortex. So these are not even simultaneously recorded neurons. So we don't even have that information. But of course, we'd like to expand the methods to be able to use also correlations, which intuitively should also give us much more information.
Okay, well, that we've run out of time for questions, but there'll be time for more discussion at the end of the 3 talks. We're going to have a period where we can have a discussion amongst all of the speakers. And we'll go on now to the second talk, which will be given by Ashok Litwin-Kumar, who is an assistant professor in the Department of Neuroscience at Columbia University. And he has been focusing on learning algorithms and their neural implementation, and not just in brains, but also in machine learning and artificial intelligence. So there's been a real breakthrough in AI, which has been really shifted from very abstract, logical, and rule-based programs to neural networks. Surprise, surprise. And the magic sauce there is learning and learning algorithms. So Ashok, can you continue now with your presentation?
Sure. Okay, so thanks for that introduction, Terry, and I'll echo Nicolas in congratulating you and the other winners of the prize. It's really a great thing for theoretical neuroscience. So today I'm going to talk about what we have learned about learning and memory from the connectome of the fruit fly. So a connectome is a map of neurons and their connections obtained from electron microscopy images, like what I'm showing on the left, which can be stitched together to form a 3D picture of a neuron and its processes. The outcome of this might be a data set like what I'm showing here, which is all of the neurons, about 140,000 of the brain of an adult Drosophila fruit fly. I'm going to talk today specifically about a part of the fruit fly brain called the mushroom body, which is a learning and memory area. So a fly, if you present it with 2 odors and you pair one of those odors with a reward, and then place the fly in an apparatus like this one, where it has a choice to either approach odor 1 or odor 2, will tend to approach the odor that was paired with the reward. And this is a simple form of associative memory that we know this area, the mushroom body, is involved in. I'll start with a brief overview of the neural circuitry of the mushroom body. So flies sense odors through receptors in their antennae, which project to a region called the antennal lobe, the first relay station of olfactory information. If you follow a projection neuron in the antennal lobe to the central brain, you'll find that one of its primary targets is this region that I'm showing in green called the mushroom body. Within the mushroom body, there are numerous neurons called Kenyan cells. Their cell bodies are at the top in this image. And they send long axons that contact so-called mushroom body output neurons, which transmit information out of the mushroom body. There isn't just one output neuron, but there are many. Here, each one is shown in a different color. And you see that the dendrites of each output neuron innervate discrete spatial regions within this structure. And we call these regions compartments. Activating different output neurons can produce different behaviors, such as approach or avoidance. But what's really remarkable about the system is that there is another class of neurons that release dopamine. These dopaminergic neurons also innervate these same precise compartments and convey learning-related signals, for instance, information about reward and punishment. And we know that these dopamine neurons are required for learning and synaptic plasticity here. And so this is a really attractive system for a theorist who's interested in kind of parallel learning systems. The analogy that we like to start with when thinking about the mushroom body is an analogy with a system that's popular in artificial neural networks, a so-called actor-critic system for reinforcement learning. So in an actor-critic systems, there's one module called the actor, whose job it is to select which action to take in any given state, and a critic that evaluates the value of any state, whether or not it's likely to lead to reward, and uses this information to update the system to take better actions in the future. And that update is schematized by the red arrows here. And there's I'll mention that these actor-critic systems are popular, both as models for learning in the basal ganglia in mammals, but also, as I mentioned, form the foundation of some modern artificial neural networks. There's a nice mapping from the circuitry of the mushroom body onto this system, where the Kenyan cells play the role of the state representation, the output neurons play the role of the actor, and the dopamine neurons are the critic, with synaptic plasticity gated by dopamine representing these learning updates. And what I'll discuss today is what we have learned about the state representation in the Kenyan cells, and then I'll turn to the actor-critic system. So, like I mentioned, the Kenyan cells are these numerous small neurons in the mushroom body that have the property that they each receive a small number of inputs through little dendritic structures called claws. 3 of them are shown in the inset. A typical Kenyan cell in the adult gets about 7 inputs from these claws. And some earlier work from Richard Axel's laboratory, actually in collaboration with Larry Abbott, had suggested that the way the Kenyan cells integrate their inputs is essentially that they select random combinations of the projection neurons, and as a result, respond to kind of random combinations of odor features. And this is a good way to produce a representation of odors that is very high dimensional, very diverse, and very combinatorial. In fact, in some theory work I did when I was a postdoc with Larry and also in collaboration with Chaim Sopolinsky, we'd argued that this number 7, the number of inputs per Kenyan cell, is kind of the optimal number under the assumption that they integrate their inputs randomly. But the diagram I'm showing on top was generated by kind of tracing a subset of neurons across many flies. And so we'd never observed in a single individual whether or not indeed these neurons have the property that they exhibit, so-called random connectivity. And we first looked at this question in a connectome that was developed in the larval Drosophila brain. So the larva is smaller than the adult. It has correspondingly fewer neurons, but the overall architecture of the mushroom body is actually quite preserved between the larval and the adult stages, making it possible to reconstruct it quickly. And this is the outcome of that connectomic reconstruction. This is every Kenyan cell in both the left and the right hemispheres of one larva's brain. There's a distribution in the number of inputs that different neurons received. I'll first draw your attention to the bottom plot here, which shows that the majority of Kenyan cells indeed integrate their inputs randomly. In fact, if you look across the left and the right hemispheres of the same organism, you'll find that they are no more similar than expected by chance, by this random drawing of connections, which I think provides pretty strong evidence that the strategy the system uses is to use random responses to produce Kenyan cell representation of odors. But like any time you look carefully at connectomic data, there is an exception, which is that there are a subset of Kenyan cells that actually have very stereotyped one-to-one connectivity with their inputs. And looking at where these neurons resided in the brain, we were able to infer that these are actually the first neurons to be born during development. And then the random connectivity is a refinement on top of that. This strategy makes sense. When you have few neurons, it's important to ensure that you sample all inputs appropriately and don't miss any. And then you can use random connectivity as an efficient mechanism to build a more diverse set of responses. So, we in fact showed that this strategy of combining stereotyped and random connectivity outperforms, in terms of the error in learning associations, a purely random model. And we took this as evidence that the strategy or the objective of the Kenyan cells is indeed to produce a diverse set of responses, and that this is done using a combination of randomness and structure. Since then, the connectomes of adult flies have become available, giving us access to a lot more Kenyan cells, and kind of the overall picture of combinatoric responses seems to hold, but there's also a lot of new things that have been discovered, such as a lot of structure in non-olfactory Kenyan cells, for instance, visual responses that we're currently trying to understand the logic of. But now let me turn to the next part of the talk and talk about the learning circuitry that listens to the Kenyan cells and transforms the representation there into actions and estimates of value. So the first lesson from the connectome about the actor-critic system is that the answer to what is the learning algorithm that it implements is likely going to be it's complicated. This is a similarity matrix of all of the inputs to every pair of dopamine neurons in the data. So every row and column is a dopamine neuron. The color is how similar are the set of inputs that they get from the whole rest of the brain. And the gray lines are the boundaries between different compartments in the mushroom body. And there's a lot of structure here, but what stood out to us the most was the degree of white space in this matrix. So white represents pairs of neurons that don't share a whole lot of inputs. And it was particularly surprising to us to see this white space because most of the neurons in this diagram are actually classically categorized as so-called reward responsive dopamine neurons. And what this tells us is that even among these putatively reward responsive neurons, they seem to be integrating very different inputs, suggesting that there may be a very high dimensional learning signal that's guiding plasticity in this system. So one of the first things you might be interested in the face of this heterogeneity is to look for particular motifs of interest, such as feedback from output neurons of the mushroom body back to the dopamine neurons. This kind of feedback would be important for being able to use internal predictions to inform the future learning. And when you trace the connectome looking for such pathways, they indeed exist. And one of the strongest ones is a pathway from neurons involved in appetitive long-term memory through a couple layers of processing back to another set of neurons involved in appetitive short-term memory, dopamine neurons. And of course, there are multiple pathways, but one such pathway stood out to us in particular, that a pathway that went through this kind of hub neuron in the middle called SMP108, which really receive the greatest degree of convergence of neural activity that is then transmitted back to the dopamine neurons. So my collaborator Yoshi also investigated the role of this neuron, SMP108, in a particular behavior called second-order conditioning. In second-order conditioning, you first pair an odor with a reward, but then you pair a second odor with the first and ask whether flies prefer odor 2 to a unconditioned odor. What's interesting about this paradigm is that you'll notice odor 2 is never presented in conjunction with reward. And so any learning about odor 2 has to occur through a kind of internal prediction of the value of odor 1 that the fly then associates with it. Yoshi found that indeed flies can robustly perform second-order conditioning. The memory score here is a number where 100 means flies always choose the conditioned odor and 0 is chance. So there's a robust memory score, and also blocking the activity of this special neuron SMP108 significantly reduces that second-order memory. Our collaborators also found that indeed optogenetically activating SMP108 leads to dopamine responses across the mushroom body compartments, which makes sense. And we were able to build a model in which we explained the dynamics of memory induced by activating SMP108 using a combination of the memory curves that had been previously described for different dopamine neurons. And when we looked at which dopamine neurons contributed to this fit, which ones were needed to explain what SMP108 does behaviorally, we found that there was a good match with those dopamine neurons that receive strong feedback from this pathway. So this is kind of a satisfying description of a precise circuit involved in second-order conditioning in this brain. But I also want to point out kind of something that tells you there's got to be more going on, which is illustrated in a movie here. And when the red square turns on, that's when Yoshi is activating this SMP108 neuron in a set of flies in an arena. In the arena, there's airflow coming from all directions. And what you saw is that the flies execute this very specific behavioral program where they turn upwind when this neuron is activated. So in addition to being kind of a memory neuron, this neuron is also involved in very specific action selection programs, which suggested to us that maybe the actor and the critic in this analogy are not so segregated as typically thought of in reinforcement learning systems. That brings me to the second part of this talk, a recent project where we've been trying to dig into the relationship between action selection and learning in the mushroom body. And this was enabled by a specific manipulation that our collaborators were able to do in flies, which is the activation of a set of neurons called moonwalker descending neurons. There's a control fly on the left and a neuron with the MDNs activated on the right, and you'll see it really causes the fly to walk backwards, which is a component of an avoidance response in flies. And so Fatima in Bertram Gerber's lab asked the interesting question: what happens if we pair an odor not with a reward or punishment, but rather with backwards walking? So give an odor and activate the MDN. There's no reward or punishment in this experiment. And when she did so, she found that flies very robustly avoid the odor in which they were made to walk backwards. That's the red curve here. She also found the reverse. There are a set of neurons called bolt neurons, which cause the fly to walk forwards, and flies prefer odors that are paired with bolt activation. But I'll talk just about the moonwalkers today. So the first question is, does this engage learning circuitry in the mushroom body? To answer that, Fatima imaged responses in dopamine neurons in the mushroom body while activating the MDN, the moonwalker neuron, and found indeed there were robust responses in punishment responsive dopamine neurons and weaker responses in reward ones, suggesting that this neuron that causes backward walking engages kind of a net punishment related portion of the circuitry involved in learning. And when we started thinking about what might the behavioral consequences or utility of such feedback be, we were drawn to a particular observation in behavioral psychology called the avoidance paradox. So animals, if they learn that a stimulus predicts a punishment, and they have the opportunity to avoid that punishment, maybe by moving to a different room in which their feet don't get shocked, they'll very robustly produce that avoidance or escape response. They'll learn to do so. And in fact, they'll continue to do so for a very long time, even well after they never get the shock anymore. They always avoid it. Or even if the shock is taken away, they'll continue to move to the other room in response to the tone. So you could say that this avoidance response is very resistant to extinction of a memory, more resistant than you would predict by a generic reinforcement learning model, where if you don't get the reward or punishment paired with the stimulus, you eventually forget the association. We hypothesized that the moonwalker neurons might be a way to preserve an avoidance memory in the face of potential extinction. And to test this, Jasmine, a student in my lab, built a simplified model of a fly navigating in a one-dimensional environment in which there were two odors, each of which activated a set of model Kenyan cells. And at either end of this linear environment, there was either a reward or a punishment, depending on the odor. The model fly could choose among three actions, either to stay where it is or to approach or avoid the odor that it currently is in. And what I'm showing here is the behavior of this model using a kind of simple reinforcement learning framework. So the y-axis is the value that the model predicts for odor 1 or odor 2. The time represents the time over many, many trials. At the end of each trial, the fly is returned to the center of the environment and gets to sample again. And the circles denote moments where the model fly received a reward or a punishment and learned something. So what you'll notice here is that the fly experiences multiple rewards, these green dots, and eventually learns a positive value for odor 1. In this simulation, it encountered odor 2 twice and got a punishment. And as a result, the value went negative. But in between those two encounters, you see forgetting, because it's not continually reinforcing the odor 2. This is an example of what contributes to this avoidance paradox. This is an animal that continually forgets this negative experience and re-experiences it. And Jasmine made a simple modification to the model inspired by what we observed in the moonwalker neurons, which is she assumed that the dopamine neuron activity isn't just related to reward or reward prediction, as in kind of vanilla reinforcement learning, but also has this additional component that when the fly takes an avoidance action, that serves as a small negative reinforcement. And this model kind of resolves the avoidance paradox. Now you'll see that the value assigned to odor 2 is retained at a negative value for a while. And so indeed, this moonwalker feedback seems to be serving as a way to preserve this avoidance memory. Now, this model makes a prediction that essentially the moonwalker neurons act as a brake on extinction learning. What that would mean is that if you simulated a network in which you condition an odor and observed a negative memory score, that model would extinguish to some degree if you presented the stimulus without a reinforcement many times. But the degree of that extinction would be greater if you didn't have intact this moonwalker neuron feedback pathway. This was a prediction that the model made, and we turned to Fatima and Bertram and asked them to do this experiment, to ask, is this extinction affected by inactivating the moonwalker neuron? And we were really satisfied to see that this prediction was borne out in experiments in behaving flies. So I think what these two stories have suggested is that rather than kind of a feed-forward circuit that associates rewards and punishments with representations in the Kenyan cells, that the mushroom body is actually a very recurrent circuit with interactions among different compartments and among actor and critic systems, and even feedback from what action was selected back to the learning circuitry, allowing for kind of moment-to-moment updates of its behavioral policy. I'll stop there, and thank Jack and Jasmine for doing much of the work, as well as a number of really great collaborators across many different labs.
Wonderful, Ashok. Thank you so much for the presentation. And I think what this illustrates is that we've made a lot of progress recently with understanding the connectivity pattern of the fly. And this is something that came out of the BRAIN initiative, which was launched by President Obama 10 years ago and has really transformed a lot of the modeling that we're doing now, because we can now use the anatomy as a foundation, and also the new tools and techniques for manipulating neurons really have come through and allowed us to test these models. So I have a quick question. Could you put your slides back on briefly? I want to go back to one of your slides, which had the air of coming in from the north, south, east, and west. Yes. It was a movie. It was a movie, and I noticed something.
Okay. You see my screen. Let me go to the movie.
Okay. So look, the air is coming in from the top, the bottom, and the right and the left. Now look where they go. They go halfway between the north, say the north and the east, as if they're trying to avoid the air, right? It's an avoidance.
Yeah, that's interesting. So I would actually need to look at the experimental apparatus here, because my understanding, I think, is that the air is coming in through the whole perimeter. But
well, it's remarkable that
it does have 4 quadrants, and they do seem to be going to 2 of the quadrants. So that's an interesting catch, and I need to maybe talk to Yoshi to understand.
Okay, yeah, I'm curious to know. Look at this. I mean, they're grouping at these 4 cardinal points. Okay, so that was my question. So let's see, Nicolas, do you have a question, or would you, Julijana? Why don't you go ahead?
I can ask, and Nicolas can maybe follow up. So I was wondering, because Ashok, thank you very much for that wonderful presentation. You focus mainly on the work you've done in the fruit fly, which is really beautiful. You have all this different modalities of data, right? Behavior, some imaging and connectomics. But I know you've also done work on the cerebellum. So I was wondering whether you could tell us what are the lessons or what are the similarities that tend to be similar, that tend to apply to both of these very different systems, one in an invertebrate and one in a mammalian circuit.
Yeah, okay. So, as theorists, we have the luxury of mixing our metaphors. So the cerebellum and the mushroom body both have this property that there are sparse connectivity onto granule cells or Kenyan cells. And so I think the way they construct this high-dimensional combinatorial representation may be shared. But then, at the output level, I drew more of an analogy between the basal ganglia and the mushroom body, because they're both dopaminergic learning systems. That being said, I think, recently there's a lot of work on interactions between basal ganglia and cerebellum. They may be really systems that work in concert a lot more than was previously understood. And one of the things I think as a field that we're still trying to work out is the nature of the error signal. That's always been the question. What's the error signal? How high dimensional is it? How diverse is it? In the mushroom body, we're finding that the error signal may indeed be not just reward and punishment, but something that's really distributed across these many compartments. And the cerebellum also has this property that there are many, many climbing fibers, and it's still unclear kind of how high dimensional is this climbing fiber error signal. It's a really interesting question.
Yeah, just to follow up with that, there's also been a real shift in understanding the signals that the dopamine cells in mammals may be carrying. It's no doubt that it's error prediction, reward prediction error, but now we know that in addition to that information is a lot of information about sensory motor signals, and that suggests that, again, it's a higher dimensional signal than just a simple scalar neuromodulatory kind of a signal.
Yeah, I agree. That's definitely been a theme in basal ganglia literature in the past years.
Okay, Nicolas, you have we have time for one more question.
Yeah, I just wanted to ask you about this, the fact that you think that so in the models you have this actor-critic separation, and you think that in the flies it's kind of more integrated, or maybe more complicated than just two separate circuits. So do you think this could have also consequences on artificial neural networks and indicate some benefits of why you don't necessarily need to separate the two?
Yeah, that's a good question, and I'm not sure I yet have a good answer for that. I think in this case, it really makes sense in light of kind of moment-to-moment learning and very active engagement with the environment. And I mean, I think you can build artificial neural networks that don't have this segregation, essentially with two different readouts of a larger network. When that is particularly important, I'm not sure I have a good guess for it, but it's a nice thought.
Okay, well, let's go on now to the third and final talk by Julijana Gjorgjieva, who is at the Technical University of Munich, and her research is all about trying to understand a real problem that brains have to solve, which has to do with, first of all, being able to adapt and learn, but on the other hand, being able to maintain a balance. That is to say, you don't want to forget everything that you've learned before, and how do you do that? And she's particularly interested in development, where new neurons and new synapses are being integrated into circuitry. And then also, this is something that's very important for evolution, which is how do you conserve energy? How do you create brains that can survive and be able to get by on relatively small amount of energy compared to what we now have with our artificial neural networks, which are reaching nuclear power plant proportions. Obviously, our brains work on much less power. So, Julijana.
Thank you very much. Also, I would like to join Ashok and Nicolas at congratulating you, Terry, together with Chaim and Larry, for your very well-deserved and fantastic achievement in winning the Brain Prize. This is really amazing, and it's also great for the field going forward. So congratulations. And thank you very much for including me in this wonderful lineup. It's really a big honor. So as you already mentioned, the work in my lab over the last several years has been on questions on how the brain of a developing organism acquires all the important connections that this particular individual will need later in life. So how do these neural circuits go from being very weakly and inappropriately connected in early developmental stages to being precisely and obviously non-randomly connected later in life to be able to execute these diverse computations and behaviors. And so we know that this is a complex process that involves many different mechanisms. Some of them are completely independent of activity. They are determined by the interactions of different sets of genes and molecules, while others depend on specific and non-random patterns of neural activity. And those are the ones that I'm interested in. So today I'll show you a few examples of how we have worked to understand how these activity-dependent mechanisms shape neural circuits in development and during learning. So typically when we think of neural activity, we think of activity that's generated by the sensory organs. So this is sensory evoked, how we perceive the world around us. However, we know that early in development, many of these sensory organs are immature. So the activity is actually spontaneously generated, but by many of the neural circuits themselves. So just to give you an example of this, here in the visual cortex, in the visual system of the mouse, of the rodent, spontaneous activity is first generated in the first organ that processes vision. And so that's in the eye, the retinal circuit, which generates these very nicely patterned events that have spatiotemporal correlations that are known as retinal waves that have been studied now for several decades by different labs, most prominently by Carla Schatz, Marla Feller, Marcus Meister, Rachel Wong, and so on. These particular recordings are actually from the lab of my long-term collaborator, Christian Lohmann at the Netherlands Institute for Neuroscience in Amsterdam. But spontaneous activity is not just seen in these primary sensory areas, it's also seen downstream. So in the visual system, it propagates in the primary visual cortex and also in higher order cortices. So here we see an example of this activity spreading completely in the absence of sensory experience in both the primary and the somatosensory, primary visual and somatosensory cortex V1 and S1, and this area in between that I've indicated here with the dashes, which is basically a multisensory area in the high order visual cortex known as area RL. And various experiments have shown that actually manipulating or perturbing this spontaneous activity can really have fundamental implications on the emergence of connectivity and eventually computations later in the life of the animal. And so we have been interested over the last several years to understand what is special about these patterns of activity in terms of their statistical structures and how could they be interpreted or used by these activity-dependent synaptic plasticity rules to organize connectivity. And so what I'd like to give you some examples of today are several studies to exactly illustrate this question. So how are neural circuits established in early development driven by these patterns of spontaneous activity? And also later on after the onset of sensory experience, how is this structured activity now from the sensory world used to either stabilize or further modify connections in these circuits? And, because we're computational neuroscientists, we can build theories and models that can basically use data sets from different experimental labs that span these different modalities. So functional data, behavioral data, connectomics data, and so on. And so what we have done in my lab is to look at this at different spatial scales. And so I'll give you an example of the emergence of this connectivity driven by spontaneous activity by these retinal waves or these spontaneous events that we see in the visual system at the subcellular, namely dendritic level. And we'll also then give you some examples of these activity-dependent mechanisms also at the level of after the onset of sensory experience, so driven by structured sensory activity. So here we were motivated by some experiments done in the lab of Christian Lohmann and others that identified very specific structures at the level of these synaptic inputs. Namely, they did 2 photon calcium imaging experiments for pairs or multiples of synaptic inputs and quantified the amount of coactivity that they exhibited as a function of the distance between the synapses. And so what they found is that in synapses that were near each other, within 15 microns of each other, tended to experience this very high coactivity or correlation, a property then they termed functional synaptic clustering. They called it functional because we don't really know what these synapses really respond to in terms of sensory features or aspects of the visual world. But what is important is that these were observed in the period when spontaneous activity is seen. So when these sensory inputs are not coming in, so the sensory organs are immature. So it's really when they blocked spontaneous activity, they saw that these clusters disappear. So this is really a property that emerges due to these correlations or structure that exists in spontaneous activity. And so with my former PhD student, Jan Kirchner, we wanted to investigate how this particular organization may merge in a model of basically a dendritic branch that's populated with multiple synaptic inputs that were initially randomly distributed. And so to understand this, we basically added these two important ingredients in the model. The first is the type of activity that these circuits experience naturally at this period, which is spontaneous activity. So here we actually used the model that was fitted to these retinal waves that appear early in development, that have the spatiotemporal statistics that we think are important for the emergence of these clusters. And then we needed a way, of course, to transform these retinal waves into some type of activations of the inputs. And for this, we used the receptive field model based on measurements of these receptive fields by a study from Sonja Hofer's lab that identified them to be basically characterized by these gabors. And so we used the center, as well as we assigned an orientation preference to each of the synapses. And so from now on, I'm going to use color to indicate the orientation preference of each of the individual synapses. And so, of course, the second and the most important ingredient here is how to interpret these activity patterns that are contained in the retinal waves, these statistical signatures, into something that could potentially give rise to order and organization. And so here, again, we were inspired by experiments that identified a specific set of molecules, in this case, neurotrophin molecules, that were found to interact in an activity-dependent manner and therefore influence the emergence of these clusters. So here, these were these BDNF and pro-BDNF molecules. And what we were able to do is write a mathematical model for their interaction and then reduce it to a more phenomenological model that we could analyze mathematically and investigate the implications of. And to our surprise, when we actually reduced that model, we found ingredients that looked very similar, like what we're used to finding in plasticity rules. We namely found a local Hebbian component that basically could stabilize synapses that are near each other that tend to be coactive. We also found a heterosynaptic component that would depress synapses that are near the ones that were coactivated if it was uncorrelated with them. And we introduced a third ingredient, which was structural plasticity, in order to ensure that the same density of the synapses was maintained on the dendritic tree. And so with this plasticity mechanism that was derived from these molecular interactions, we were able to investigate the type of structures, the type of organization that emerges of the synaptic inputs on these dendritic trees. And so what we found indeed was that the synaptic inputs can organize themselves into these clusters, and we can visualize them, or we can summarize them in the same ways in the experimental data by looking at the correlation as a function of distance. And this is what we see in the cyan curve. And when we stimulate the model with just completely random noise, we completely abolish, or we don't see the clustering relationship. And what was interesting to us is that even though this was a developmental model that was actually tuned on mouse data, we were able to find a wonderful matching in terms of this clustering that was observed in the visual cortex in the ferret by the Fitzpatrick lab. But actually, here the functional correlations emerge because the receptive fields actually line up to really share the same orientation. So as we can see here in the schematic, but if we look at the deeper in the data published by other labs, it turns out that the mouse actually in adulthood does not really show this orientation clustering. Rather, the mouse shows a different type of clustering of receptive fields that is due to overlap. So we can see it here in the schematics below of the Gabor receptive fields. So you see here that the positive regions shown here in red really overlap, even though the receptive fields that are nearby are completely not aligned in terms of their orientation. And so it turns out that this overlap is actually sufficient to generate this functional overlap that is necessary to generate sort of this clustering conclusion, this clustering type of organization, even though these receptive fields are not, when they're nearby, they're not actually similarly oriented according to orientation. And so our model could even be applied beyond these two species. I forgot to mention, but the factor that we included to separate what was different between the ferret and the mouse was actually the sizes of the cortices. So the ferret cortex is about 30 times bigger in surface area than the mouse, and therefore has a much bigger amplification factor. And so including such size arguments, we could actually predict whether the cortices of macaque or cat or rabbit would actually have orientation or overlap clustering. And we could further extend these models to investigate also the emergence of global organization. What I mean by that is the organization of synaptic inputs on the entire dendritic tree of the neurons, as opposed to just these small dendritic branches. But I'm going to switch gears a little bit rather than showing you this global organization and discuss how we are using similar flavor plasticity rules of this sort of Hebbian flavor that require the coincident activation of pre and post-synaptic neurons to give rise to potentiation, to give rise to the emergence of other structures, not now at the subcellular, but more at the circuit or the network level. And so in this case, we were interested in how this self-organization emerges, not just for the sake of the emergence of certain structures or ways in which these neurons or synapses are organized, but rather in order to produce a certain computation. And the computation we had in mind here was a computation known as response normalization, which basically refers to the sublinear response to a summation of two stimuli relative to when the two stimuli are shown on their own. So these are experiments that were, in this case, done in the cortex of the cat, in the visual cortex of the cat by Laura Busse, but also have been repeated also in the rodent cortex. And so when one looks at the responses of a population of neurons in the visual cortex to oriented gratings, in this case, the horizontal or the vertical, one can see basically population curves that are peaked at the particular orientation that is shown. But interestingly, when two stimuli, in this case, these two orthogonally oriented gratings are shown together in the form of this plaid grating, the response of the population is actually much lower than the sum of the individual responses summed together. So it's basically a nonlinear computation that means that is this sublinear summation. Moreover, when one of the stimuli is shown at a lower contrast, then the network does winner-take-all competition in the sense that one of the stimulus that is shown with the higher contrast is really amplified at the expense of the other one that is heavily suppressed. And so the question is, how does this response to normalization emerge in biological neural networks? And the answer to this is already known. So this, we didn't come up with this. This is the so-called or well-known stabilized superlinear network, or the SSN, that is basically a network comprised of excitatory and inhibitory units that are very strongly recurrently connected with each other, in the sense that the network is inhibition stabilized, in the sense that if you don't have inhibition, the network will blow up. And a second feature that this network has, besides this inhibition stabilization, is the fact that it has this nonlinear activation function for the individual neurons that follows this power law function. And so this network was studied by Yashar Ahmadian, Ken Miller, Dan Rubin, and so on, who basically, the network, in addition to this recurrent connectivity, also receives this feed-forward input that is tuned to a specific orientation. And so when you show a particular orientation, you can see the same type of single bump response as we saw in the experimental data sets. And what they found, indeed, is that this network can generate this type of response normalization, in the sense that if you show the two stimuli one at a time, you see bigger responses than if you show them together. So the summed response is smaller than the sum of the individual responses. And the network also does this winner-take-all competition, so it amplifies the stronger stimulus. But these models had hardwired connectivity. So they looked at parameters which in the visual cortex may have been measured, connectivity parameters, but it was not clear how this connectivity may emerge, especially for circuits where we may not have these kinds of measurements. And so what we wanted to do here was to actually look at the type of activity-dependent plasticity mechanisms that could give rise to the emergence of this type of connectivity that can generate these nonlinear computations like response normalization. And so here we took a fully plastic network of excitatory and inhibitory neurons that receives feed-forward inputs from these units that are, let's say, orientation tuned, that are orientation selective. And we allowed all the connections in the network to be simultaneously plastic, the feed-forward as well as the recurrent, excitatory, and inhibitory. And rather than investigating specific plasticity rules, which we usually do, so for example, in the subcellular work I showed you earlier, we derive the rule from the known molecules that are present in the system. Here we wanted to be agnostic and use the simplest rule we could think of, which is the vanilla Hebbian rule, that basically says that the connection between two neurons will be potentiated if the activity of the two neurons is coincident. So if they're both active at the same time. So fire together, wire together. But as we know, this classical Hebbian learning suffers from the problem of runaway dynamics. The more activity there is, the more the synapses potentiate and activity grows. And so my former PhD student, Samuel, had the idea to introduce a normalization, so what he called stability through competition for limited synaptic resources, whereby, so it's effectively like divisive normalization. So if one synapse, in this case, the one with the two stars, becomes potentiated due to Hebbian LTP, then this normalization, so this competition for synaptic resources would scale both synapses down while preserving their relative strengths. What the insight that Samuel had was to actually introduce this normalization separately or independently for both excitatory and inhibitory synapses. And this was based on some experiments, legacy experiments, I would say, by Bourne and Harris, that measured exactly and showed that this total synaptic area is preserved after LTP induction for both excitatory and inhibitory synapses. And so to analyze or to investigate the plasticity in the entire network, what we did was to first focus on the individual connectivity motifs in the network. So we first had, we had a neuron in the network that had a flat, basically untuned tuning curve from the inputs. This is shown here in the blue line. And then we had a lateral neuron that was recurrently connected to the dark blue neuron here. This is the one shown in the light blue that actually was tuned to the inputs. And so when we allowed this plasticity, both feed-forward and this recurrent connection from the tuned to the untuned neuron to be active, we saw that basically the tuning curve of the untuned neuron really converges or becomes the same as the tuning curve of the tuned neuron. And so what this means is that this excitatory influence is very attractive. We can do the same experiment now by looking at a motif that includes an inhibitory neuron as opposed to a lateral excitatory neuron. This is the one shown here in the red. And in fact, we actually have several of them that all have tuning curves that are highly selective. These are the ones that are shown in the red. So you see they all look like bumps that are distributed in orientation space. Now, if we allow these inhibitory neurons to have their influence on our downstream pyramidal neuron in the dark blue that is untuned, we see that the effect of the inhibition is to actually pull away or repel the tuning curve that emerges for this new excitatory neuron away from the ones that exist for the inhibitory neurons. We can continue making the circuit more and more complicated by adding more neurons, more connections, and therefore more connections that are plastic. And so here we add two excitatory neurons that have feed-forward tuned connections. So the two blue curves that we see are the tuning curves for the feed-forward connections. But the neurons themselves are plastic and connected with these recurrent connections. And the intuition that we got from the motif above is that the influence of this excitatory recurrent connectivity would be attractive. And so indeed, when we introduce plasticity here with this competition for resources, we see that the two tuning curves basically become attracted to each other and become the same. And we can repeat the motif, but now replace the excitatory by inhibitory neurons. And what we see is that the tuning curves, the feed-forward tuning curves of these inhibitory neurons become decorrelated. So basically, we can now combine this in a large network where all the connections are plastic and investigate what happens. And so indeed, what we see is that these inhibitory interactions can be used to decorrelate the excitatory tuning curves. And the reason that this happens is that the increase of attraction that we see due to an additional excitatory neuron is always balanced by a simultaneous decrease in the attraction by an additional inhibitory neuron. And so if we look now at the fully plastic network and we focus on the emergence of the change in the connections according to this plasticity rule, both in the feed-forward and in the recurrent part, what we see is indeed emergence of structure at both levels. First, at the level of the feed-forward connectivity, even though we start from a network that is initially flat, where the tuning does not exist. So I'm going to change again and show you that again. So we see flat tuning curves, and the tuning curves become organized. You see that initially the inhibitory ones are the ones that decorrelate first, and they're the ones that then impose the influence so that the excitatory tuning curves also decorrelate. But we also see interesting or very organized structure emerging at the level of the recurrent connections between the excitatory and the inhibitory neurons. And so here we line up in this connectivity matrix the excitatory and the inhibitory neurons according to their orientation preference in terms of the feed-forward connectivity. And so we see an excitatory to excitatory matrix that looks very beautifully structured in terms of its, and the fact that it's block diagonal. So that means is that neurons that share similar feed-forward preference actually end up becoming more strongly connected to each other. And this also applies to the other types of connections between the excitatory and the inhibitory neurons, as well as within the inhibitory neurons as well, which very nicely is in agreement with experimental data. So our models' connectivity are in good agreement with experimental results, but what about the computation? So we started by saying, can we get an SSN by basically looking at the plasticity mechanisms that generate the appropriate connectivity? And so to do this, we did this experiment exactly like in this response normalization computation. So we presented two different stimuli, in this case, for example, two orientations, one at a time, shown here in the orange and in the green. And when we showed them together, we saw that the response of the network is actually lower than it would be when we sum the two individual responses. So indeed, the network can do this nonlinear computation. And similarly, the network can also do this winner-take-all competition in the sense that it will amplify the stronger stimulus at the expense of weakening the or suppressing the weaker stimulus. The networks can also do other computations that I don't have the time to discuss right now. For example, it can do surround suppression, and it can do surround suppression in interesting ways depending on the statistics of the stimuli that are used to train the network. And so what we showed now was that indeed we can get this simple Hebbian rule without any big assumptions, plus this competition for synaptic resources to give rise to decorrelation of the feed-forward tuning curves and this like-to-like connectivity that emerges between the recurrent connections. And without really having to fine-tune the connections, we basically get a network that can do this interesting computation, which is response normalization, surround suppression, and so on. But in the last few minutes, I wanted to address the question of, well, what about the actual signal that neurons use to communicate with each other? So far, we mainly focused on rates and how using these rate-based models, one can investigate what are the types of structures that emerge and computations that emerge in these networks. But neurons actually use spikes to compute, and they also use these spikes to modify the connections between the neurons. And indeed, various experiments have shown that, and Nicolas alluded to this, showed this in his talk, that the precise pairing of spikes can, during plasticity induction protocols, can really affect the type of plasticity that is observed and measured at individual synapses. So these are experiments that were done in layer 5 pyramidal neurons in the auditory cortex in the Fromka lab that did a kind of a classical pairing paradigm where they showed pre- and postsynaptic spikes at both excitatory and inhibitory synapses into a given pyramidal neuron in the auditory cortex. So when they did it for excitatory plasticity, they found the classical, let's call it classical STDP, spike time independent plasticity curve, also measured by Bien Pu, Dan Feldman, Gina Turgiano, Sjöström, and others. But they also actually measured an inhibitory plasticity rule that looked different than the excitatory one in the sense that it sort of induced potentiation or depression without caring about the order of the pre and the postsynaptic spikes. And so in some recent work, we have started to ask about the nature or the properties of these types of inhibitory STDP rules and the types of connectivity structures and motifs they can give rise to in spiking networks. And so this is recent work that was done by my current postdoc, Dylan Festa. And so what we here wanted to investigate first at the level of small motifs comprised of an excitatory and inhibitory cell, if we just fix the excitatory to inhibitory connection and allow the inhibitory to excitatory connection to be plastic, what are the types of motifs that can emerge? We were investigating the emergence of motifs like blanket inhibition, where the inhibitory neuron would potentially provide inhibition to all the excitatory neurons in the microcircuit around it. The emergence of a mutual connection, where the inhibitory connection would be reinforced to the excitatory cell from which it receives a connection, and lateral inhibition, where the inhibitory neuron will project to excitatory cell from which it does not receive connection from. And so to do this, we wondered, well, first of all, what is the type of inhibitory rules that we should use? For excitation, people have measured a variety of rules, and Nicolas showed several of them. But for inhibition, we have this measurement, we have maybe a few others, but definitely not a whole array, a whole family of rules that we have for excitation. And so we wanted to basically investigate potentially different plasticity rules and how they might be influencing the emergence of these different motifs that I showed you a moment ago. And to do this, we used a framework that allowed us to decompose the contribution of the plasticity to this inhibitory to excitatory synaptic connection. Just like people like Richard Kempter and Wolfram Gerstner did many years ago for excitatory plasticity. So we allowed plasticity to be written by a contribution of terms that involved presynaptic, postsynaptic firing rates, a term that included the contribution, mainly the product of the pre and the postsynaptic firing rate. So this is like a typical Hebbian term that needs both pre and post. But then we also allowed introduced a term that uses the spiking correlations specifically that are integrated through the STDP kernel. So this function, the shape of this plasticity rule, to contribute as well. And so what we found is that we first considered the family of plasticity rules where we allowed certain terms to be excluded. For example, this correlation term to be excluded. We call this rate-dominated learning rules. And just a small but important note that these are the types of plasticity rules that were used by Tim Vogels and Henning Sprekeler together with Wolfram Gerstner to basically investigate or to demonstrate that these rules have very powerful homeostatic properties in the sense that they can stabilize firing rates in the network. And we found that indeed, if we allow these learning rules to operate in our little motifs, what we see is indeed stabilization of firing rates. So these two blue curves are the firing rates of these two excitatory neurons in our motif. And the two inhibitory connections become of very similar strength. And so this is interesting because it basically means that the network is indeed doing homeostasis by basically generating blanket-like inhibition. So this inhibitory neuron connects to all the excitatory neurons in the network. We also studied what we found very interesting is then we studied correlation-dominated learning rules. And so in these learning rules, we wanted to eliminate the contribution of the area under the curve of the plasticity rule because that seems to have dominant effects and is determined very strongly by basically the area under the curve of the STDP kernel. So we took here STDP kernels that have area under the curve equal to 0. So the blue and the red region here cancel. And these are, in this case, the term that dominates is this correlation term Q. And what we found here is very different behavior. Namely, we call it, it looks like winner-take-all dynamics in the sense that only one of the inhibitory connections in this motif becomes stronger, and the other one basically becomes weaker and completely depresses. And in the case when the plasticity rule has this symmetric shape, the motif that is promoted is this mutual connectivity motif. On the other hand, if the rule is antisymmetric, we found that the rule still does winner-take-all competition, but now the motif that is promoted is actually the one that is a lateral inhibition. Okay, and so we are now investigating basically what happens in networks where we can include both of these plasticity rules. So we add two populations of inhibitory neurons and we're including both of these plasticity rules. We're investigating the emergence of structure. So for example, we see the emergence of effective Mexican hat connectivity that can generate interesting computations. For example, can it generate surround suppression? It can actually generate correlations and sort of spontaneous activity that tends to propagate over very long spatial scales. And so we're really studying what is the potential of these different types of plasticity rules to generate different connectivity motifs in these networks. And at the very end, I just like to summarize by telling you that what we have seen is several examples from the work in my lab that have investigated how activity-dependent plasticity can shape organization, both at the subcellular and the cellular scale, and can give rise to the emergence of these different types of connectivity motifs, as well as computations. And at the end, I would just like to thank my group for all of their amazing work, because they're really the ones that they did this work, and I'm happy to take questions.
Wonderful. So that really covers a lot of different models and different observations. So you mentioned the legacy paper by Bourne and Harris. So I collaborate with Kristen, so she'd be very pleased to hear that. And you know, Bourne died tragically in an automobile accident, and now there is a prize for anatomists that is in her name, which I think is very appropriate. And you know, the clustering is very really very interesting, both in terms of mechanisms and also function. But in addition to you know, activity-dependent clustering, there's also intrinsic homeostatic mechanisms. And I wonder if you've taken a look at that and seen if that is also might be an important addition.
Yeah, I mainly made the talk today about how we can study plasticity at different spatial scales. So focusing, of course, on the subcellular, maybe in the circuit, and we also have some work even at the interactions between these different brain regions. But of course, the other thing we can do is look at these at different temporal scales. And so I think all of us have looked at this, right? So this is really the beauty of what we can do with these cross-scale models. So mainly what I showed today was indeed long-term plasticity, so mechanisms that operate on the order of, let's say, tens of minutes to hours. But of course, there's also short-term dynamics and short-term plasticity. And on the other hand, there's more homeostatic mechanisms that operate on longer time scales. And they're, of course, important. And I think ultimately brain circuits use a combination of all of them. And we have looked also at homeostatic mechanisms. It's just that I think when you put all of these different timescales together and all of these mechanisms that operate at the different timescales, of course, they all play different roles. So classically, people think homeostatic mechanisms are maybe important for stabilizing dynamics or learning over really, really long timescales. But more recent work shows us that, well, yeah, they can also happen also at shorter timescales. So I think it's important to consider them. These homeostatic mechanisms can be diverse. So they can be intrinsic excitability, they can be mechanisms of synaptic scaling, they can be heterosynaptic mechanisms that operate at synapses where plasticity is not directly induced. Yeah, that's the beauty, I think, of how they work together. Yeah.
Okay, well, you clearly have a wonderful set of questions ahead that you can continue to attack. Okay, well, one more question. Anyone would like to add another question?
I can ask a question. I had a question about the second part. I think you really nicely showed the importance of inhibition in these recurrent networks and creating a set of responses that span the stimulus space. I'm wondering, is there any setting in the model where excitatory interactions are very important?
So that's a good question. I mean, so I was expecting that you would ask me about the stimulus statistics, because what our goal was initially was to get this decorrelation of the tuning curves. And what we saw was that when excitation is too strong, then the tuning curves tend to cluster together. And this is also what you see actually when you bias your stimulus statistics. So when here we mainly show our uniform, every orientation is equally likely. But if you then bias certain orientations, or if you increase these excitatory interactions, so you allow them to dominate, maybe happen at a faster speed than inhibition, then you tend to see clustering of the tuning curves. And initially we found this very interesting I think my student Samuel found some, I mean, of course, we have the clustering in terms of the cardinal, the overrepresentation of the cardinal orientations in visual space. But I think he also found, with respect to other features, some clustering, like in terms of maybe color preference and so on. But yeah, other than saying we see clustering, I'm not sure what, we still have to think more about what this clustering could mean, or relate it more closely or more directly to experimental findings where this clustering is seen.
Wonderful. Okay, well, now we have an opportunity to open up our discussion. And I want to preface the discussion with a little history for those of in the audience who may not know that the Society for Neuroscience was founded 50 years ago. And I went to some of the early meetings as a graduate student. And, you know, it was very daunting because, you know, there are thousands of posters out there. And I remember I was a physics graduate student and there were almost none. I mean, really a very sparse set of posters, maybe one or two, that had anything to do with modeling or theory or computation. It was because it wasn't really at that point a part of neuroscience. I shouldn't say that. There were some pioneers out there, Jack Cowan and others that clearly they were the one or two posters, right? But that's all changed. And I think that one of the reasons has to do with the fact that neuroscience was data poor back then. That is to say, you know, techniques for recording from neurons one at a time really limited the data sets. And also we didn't have anatomy really to help in the cortex. And so now we've shifted to data rich. We have these enormous data sets, you know, terabytes of data per experiment. And now the real problem is how to analyze it and how to reduce it to the point where we can with theory we can explore it and with new concepts and new ideas. So that's where we are today. And the segue here is the recent Nobel Prizes that were given for artificial intelligence. So the physics prize was given to John Hopfield, my former thesis advisor at Princeton, and also Jeff Hinton, my former collaborator. So I was very pleased that very well-deserved prize. But isn't it interesting also that the three of us who got the Brain Prize also had backgrounds in physics, right? And I think all of you have had some background in physics. Is that right? I know that Nicolas and Ashok, okay, and Julijana. Okay, well, how do you account for that? How do you account for the fact that physics has such an important role in at least the pioneering and now the younger generation coming up? Does that have anything to do with the tools and the approach that physicists take? And let's just start with Ashok. What's your take on this?
Yeah, it's an interesting question, and I think it comes both from kind of culture. You have leaders who made the transition from physics to neuroscience, and they kind of created trainees and established a community. And I think also when thinking about large data, and that includes both connectomics and neural activity measurements, there's a lot of disorder, and the kind of statistical approaches that statistical physics has for dealing with that disorder have just turned out to be really useful in understanding a complex system with a bunch of interacting elements with disordered interactions.
Yeah, I think that also motivated John with his Hopfield model, which was taken from condensed matter physics, which was spin glasses, right, which have both the excitatory and inhibitory interactions between spins. So this is really, I think, a good source of tools and techniques. But so, Julijana, what is your take? I think we could go further with this, right?
Yeah, well, so actually, I'm a mathematician by training. I didn't train as a physicist. My undergraduates is in math, and also when I did my master studies, I studied applied math. I was in applied math and theoretical physics in Cambridge. But then after my PhD, I went to Heim, and so this is when I think I learned a lot of my physics tools and techniques, and got maybe to think a little bit like a theoretical physicist and working on problems in theoretical neuroscience. I mean, I think one of the advantages of having this training in theoretical physics and applying it to neuroscience is just the ability to conceptualize problems. I think it's the ability to think from the complex biological data, which I think is overwhelming when you switch fields and you're reading and understanding or talking to somebody about data. So one is the conceptualization, so being able to make assumptions and reduce the problem into something that is mathematically tractable. And then the second thing I would say is then having the toolbox to study this problem. So I think the second part was actually difficult for me, coming from math into the theoretical neuroscience field and working with Heim, because as a mathematician, I was always trained to calculate things precisely, proving things. And there was always a very clear way to get from A to B. And in theoretical physics, that's not the case. You have to expand things. You have your perturbations, and you have your, reductions, and you have your tricks. And so I think now I see those as sort of the two, in my mind, the two important insights that maybe theoretical physics brought into neuroscience.
I can fully appreciate the difference between the mathematical approach, which tends to be very crisp, and the physics approach, which is has an empirical part of it, which is really dealing with data. I guess mathematicians do have data, but it's more computational data than it is physical data from nature. But in any case, absolutely right. By the way, I gave a talk. It was a meeting, National Academy meeting, which was held in 2017, and it was the mathematics, or it was the science of deep learning, and it was organized by mathematicians, and mathematicians, of course, were interested in because these deep learning networks seem to go against all the received wisdom in both statistics and in optimization theory, in terms of they shouldn't work. There's just too many parameters. but the beauty, I think, is that I gave the kind of the closing address, and I pointed out, look, these are very, very complex models, but they're just functions, mathematical functions, and who knows how to analyze mathematical functions better than mathematicians, right? So we should have access. We have access to every little input and connection and unit and activity pattern. So we'll figure it out. Mathematicians will, I should say, will eventually figure out what's going on. But the other thing, and this is actually now, coming from my physics background, a lot of mathematics have come from people who were analyzing data in the world, and two examples are Gauss, who was analyzing. He was doing surveying, for his living when he was young, and he made measurements over and over again and said, hey, this is interesting. It's not the same every time. And lo and behold, there's a beautiful theory underlying that. And the other was Joseph Fourier. So he was trying to solve the heat equation because he was up in the mountains, and came up with this idea of a series, right, Fourier series, and that created a whole new branch of mathematics, functional analysis. So beautiful theories come out of physical, literally empirical data and physical measurements, and that's continuing now, because deep learning is totally developed by engineers, empirical. Well, I shouldn't say totally, because obviously physicists played a role there too. But it was basically empirical in a sense, people doing simulations, and now that is really having a huge impact on what the economy and the public. So this is an interesting time that we're living through. So Nicolas, now what's your take? And could you put your image up so we can see your beautiful face?
Well,
I can see it.
I can see myself, but
oh, okay, okay.
I'm not sure how
I can't, but
I'm not sure how I should position myself. But
no, it's okay. I my screen doesn't show it, but go right ahead.
Right. Well, I mean, I think the Hoffman model is a great example to show how the physicist approach is very powerful. Also in neuroscience, I think physicists are trained to build mathematical models that describe natural phenomena using kind of a minimal set of like a kind of minimalistic description. And try to and are good at, I think, identifying what are the crucial variables that control the dynamics of a system, and what are the variables that maybe you can neglect, and I think the example of the Hoffman model is a great example of a model that abstracts a lot of biological details, but really it really captures the essence of the phenomenon, and also it has shown a remarkable robustness to variations in its details. So I think it's a perfect example to show why I think physics approach has been successful at modeling systems in neuroscience.
Let's make another comment on that. I'm wondering what you all think, but as someone looking back at those papers from the 80s, it strikes me as almost the problems were more crisp back then. You had this very clear model. There were clear questions to ask about capacity and questions to formulate and techniques needed to get the answer. And what's kind of nice is it seems like as time goes on, the questions have gotten less crisp. We've engaged with the data more and more and realized how messy the biology is. But because the community existed and physicists were drawn in, it's kind of continued even in an age of not even knowing quite what the quantity we're trying to calculate is.
Well, I think Julijana really put her finger on it when she said that it's not just the tools, it's also the conceptual framework. And here I think is where biology really departs from physics. And it took me a while to understand the culture in biology. So if you're doing a physics experiment on electrons, it doesn't matter what lab you're in or anywhere in the universe, because all electrons are the same. But in biology, there's nothing that's the same. Every individual has a different genome, and every experiment you do, it has different results depending. You have 10 rats, you do 10 experiments, and you get different results, and you have to somehow figure out what's common from the diversity. And so biology is all about diversity, and that really has a different consequence, both in terms of how you design experiments and also how you analyze the data and so forth. And I wonder how you guys have dealt with that problem of the issue. It's not a problem, but it's an issue. It's a very interesting issue because evolution all depends on diversity too, right? We wouldn't be here without diversity. So Julijana is shaking her head.
Yeah, how do we deal with the diversity and the heterogeneity? I mean, I think we've gone through a phase where, clearly the data we had several decades ago was individual neurons that were recorded over many trials, and we were asking questions about, signal to noise ratio and reliability, all at the level of single neurons. And now that we have techniques that allow us to record from many neurons at the same time, that allow us to look, at behavior and really find one-to-one links between certain neurons or circuits and that behavior, and also with connectomics. And now we also have connectomes for which we have functional activity. So really the data is becoming significantly more complex and more involved. And I think with this growth, we have also partially seen a little bit of the feeling of, oh, well, let me try the same algorithms, let me try the same decoding, but now instead of 10 neurons, I have 10,000 neurons. And I think, this has been kind of useful because we clearly see improvement of all the decoding algorithms. But what we're lacking is, with these types of approaches, is of course distilling like, well, no, we need a different way to look at what 10,000 neurons are doing than, you know, what 10 neurons were doing by just improving the decoders or adding more elements to the decoders and so on. Yeah, I mean, and this is really happening. I mean, more and more people are embracing this multimodality of the data sets and the heterogeneity. And we're also trying to do it. I mean, everybody's doing it at different levels of success, I would say. We all try to still find some way to simplify things, to build models that we can study, right? I mean, what did George Box say, right? Every model is wrong, but some models are useful. So we try to find the right level to reduce the data or extract the parameters that we need to answer the question that we're interested in. And I think, yeah, probably the future of how to continue looking at this diversity is to continue to distill it to the questions that we're trying to answer it. I'm not sure, maybe I can hear more from Ashok and Nicolas to think a little bit more about refining my answer, because it is really, sometimes it can be overwhelming, but that's interesting too. It's exciting. I think it's a really special time to be in the field.
Okay, others?
Well, I mean, I think one of the big challenges currently is really to understand this whole diversity of cell types, for example, that you see in practically any brain structure, and that's something that theorists, I think, have ignored for a long time, but now it's hard to continue ignoring. We have to, but I think some people, I mean, Ashok is kind of diving really in with really taking connectomes very seriously and studying really real connectomes. But I think other people are also starting to incorporate more cell types into models and try to understand the impact of these cell types. Like, for example, I think if I understood correctly, your last slide, Julijana, you have different subtypes of inhibitory neurons. And I think actually theory can also have a role to understand what are the functional roles of these different cell types. What are the functional roles of this diversity? And I think there are a few cases where we have made some progress, for instance, of understanding diversity of retinal cell types using, information theory principles, while in some cases it's actually beneficial to have multiple cell types as opposed to one. So I think there's hope that theoretical tools can more generally allow us to understand why we have so many cell types and just one, like in the Hopfield model. Or
yeah, that's
that diversity can suggest new models and new questions. Like you look at the connectome, it's very much not a Erdos-Renyi graph. It's very much not a small world network. So, what is the correct statistical model for capturing at least what diversity seems to be essential? It's an interesting question.
Right. So this is very interesting. Here's another difference between physics and biology. So in physics, there are a few parameters, the fundamental constants of nature, right? And what you try to do is come up with an equation that, like electricity and magnetism, Maxwell's equations, that summarize all the experiments, right? That's the goal. Well, in biology, there are millions of parameters, billions of parameters, right? 10 to the 14 synapses in the brain, and they're plastic. They change with experience. And, that throws you into a completely different view about what the problem is. It is a complexity problem, but I think it's actually a conceptual problem that we have, trying to understand how is it that we can talk with each other, understand each other, even though all the parameters in our brains are going to be different from each other, right? I mean, somehow there has to be something conserved that makes it possible for the brain to maintain stability over long periods of time, and for us to be able to form societies and be able to create science. This is a big mystery to me.
I mean, maybe I can say just a small note. I mean, I also trained with Yves Marder, and I was for a year and a half in her lab, and I was, of course, influenced by the notion of degeneracy. I mean, of course, Eve promoted this idea for the stomatogastric nervous system, and this is a very simple system where we know what the neurons are, we know what the connections are, we know what the type of output activity the circuit is supposed to produce, but yet we do also know that there's multiple parameters that can be orders of magnitude different and still produce the same function. And I think we're really realizing this, of course, for everything now, for all the circuits that we're studying, also in mammalian areas as well, and computations and behaviors. And in the end, I think the common parameter is, as you said, the function, the behavior. So we are indeed all able to talk. So that what is preserved is the ability to talk, but then the parameters that implement that particular function can all be different. So there is indeed a lot of degeneracy in biology that we're not used to seeing in other fields. And I think it's being discovered more and more. I mean, I could maybe mention, people, I think Jakob Make, for example, has developed this very interesting toolbox known as simulation-based inference that actually uses deep learning we're using it quite a bit actually, because it allows you to find parameters of solutions to mechanistic models, and together with Tim Fogels, they've implemented it to also optimize learning rules. So you can really get like thousands of plasticity rules that can generate certain functions, and you can filter them out. So they even have like a filtered version of SPI where you say, okay, if I say that I want my network to fire between 5 and 50 hertz, these are the plasticity rules. But if I also want my network to generate or separate novel from familiar images, then you get further and further refinements of these parameters. And yeah, I think this degeneracy concept, I think, is really fundamental to biology that I think is not so in physics.
So Eve Marder had a beautiful set of speakers in a symposium at the last Society for Neuroscience meeting that was held in October, just well, actually, it was just a couple of weeks ago. But one of the talks was given by somebody who studies bacteria, cell division in bacteria, and it turns out that the transcription factors there are also heterogeneous, but somehow they do divide, but there are a bunch of different variables that have to be coordinated, inside the cell. So it extends all the way from a single cell all the way up to the whole brain. And by the way, what do people think about these drifting representations? So this goes against the label line hypothesis that I grew up with, where the neuron has some feature that it is determined, early on, and then it fires when that stimulus is present. And now it's not true. How are we going to deal with that?
Well, I think it's not that surprising in a way because the system is so plastic. So I think it's kind of in a way to be expected if you have plastic networks that receive. So but in these experiments, what they show is that basically between like 2 different days, you see that the representation has shifted. But in between these 2 different days, presumably these networks have undergone plasticity due to other things that the animals have done, and then in that sense, I don't find it so surprising. Of course, if it depends also on the magnitude of the drift. If the representation becomes totally uncorrelated after a few months, then that is surprising. Given that it's driven by the same inputs, then that is more surprising.
Well,
maybe
go ahead.
One can think of it as moving through this space of degeneracy, so that drift still allows you to move to these different solutions.
Yeah.
Yeah.
Okay, that's a good connection. And Ashok, what about flies? Do they have drifting representations?
I think as a former kind of cortically focused person, it took me a while to accept how hardwired the majority of the fly's brain is. It's actually very specific, and most neurons can be given a name, and you can find that exact same neuron with pretty similar connectivity in the brain of another fly. What's interesting is that those regions with the least degree of stereotypy are also those regions that are involved in learning. So the Kenyan cells are the least stereotypical neurons in the fly brain, and also neurons in the central complex that have this plasticity to anchor a heading direction to a landmark. Those are also kind of non-stereotypes. So actually, I really agree that drifting representations and non-stereotyped connectivity seem to go hand in hand with learning.
Well, okay, I think we've run out of time, but this has been a fabulous set of talks. Thank you so much. And also, this discussion could go on forever because we have so many interesting topics and issues that could continue. And just, for the audience, just to know that, this field, this is theoretical and computational biology, is really become a major part of neuroscience, and there are many really brilliant young researchers coming up. That we have 3 here, but it's really wonderful to see how it's blossoming and how it's integrating, and this is really an exciting time right now to be a neuroscientist, I think, and it's happening also at the cellular level, molecular level, development. I mean, what the tools we have now are just fabulous, and we're getting answers to these questions. Okay, well, thanks very much, everybody, and good luck.
Thanks.
Bye. Thank you.